Hi guys. This time we are gonna look at a paper of two questions for 2020 October and November. It's 21 version. There are 22 and 23, but not 32, but 23. But we're gonna look at 21 because it's the basic one. I mean, it's not really the basic, but I'd say all the questions, majority of the questions for 21, 22, 23 are the same. There's some differences, like some different questions are in 22 and 23 as well, but we'll cover that later on. And meanwhile, we're just gonna concentrate on doing 21 because, yeah, most of the questions are same anyway. So let's get started. Radio. The number one. Hedgehogs are mammals. Touching a hedgehog causes it to roll into a ball to protect itself. Which characteristics is it displaying? So we can see that the hedgehog senses the touching. So sensitivity is one of the characteristics, and it rolls into a ball to protect himself. So therefore he or she or it made a movement. So the movement is the answer. Hence the answer should be D. By now you guys should remember 7 characteristics of living things. So we call it Mrs. Gran. So movement, respiration, sensitivity, growth, reproduction, excretion, and nutrition. So there are 7 characteristics of living things, and movement and sensitivity are 2 of the 7 characteristics. Okay, so this is a very basic thing, so you guys should know by now. Alright. Number 2. What are features of the leaves of a plant that is a dicotyledon? The dicotyledon has 2 cotyledons. So if you look at the seeds, some of the plants have seeds that have 2 cotyledons. So cotyledon is simply put, it's like a food storage. So it provides food for the embryos inside the seed, and the embryo later develops into a plant. So this part is called shoot, and this part is of course the root or roots. Okay, and this part is called embryo, which consists of plumule and radicle, but we'll talk about that later. Okay, we'll talk about that in plant nutrition or plant transport part. So the embryo has to receive food in order to grow, and that is given by, or that is provided by, cotyledons, and actually also an endosperm as well. But at this stage, you guys don't have to know about the endosperm. You guys just have to know about the cotyledon. So some of the plants that are that have 2 cotyledons are called dicotyledonous plants, and they have a few things in common. So things like plants that are dicotyledon are they have broad leaves, and their veins inside the leaves are net-like structure. Whereas monocotyledon or monocotyledonous plants have leaves. They're very narrow. Their widths are very narrow, and they have veins which are parallel. Whereas dicotyledons have wide leaves and they have net-like veins. So, broadleaves, yes. Parallel veins, no. Hence, the answer should be B for this one. Okay? Number 3. The photomicrograph shows a cell from a type of aquatic plant. Each part labeled on the photomicrograph indicate that this is a plant cell. As we already know, the plant cell has cell wall and large vacuole, as well as chloroplasts. So from the photomicrograph here, I can see that the biggest circle is nucleus, and then the smaller ones, these ones, are chloroplasts. And Q, of course, is the cell wall. And P is rather a bit confusing. It can be quite confusing, but it's actually attached to the cell wall here. So it must be cell membrane. Hence, so chloroplasts, cell walls, are the characteristics of plant cell that distinguishes from animal cells. So answer should be Q and S, D. Number 4. The diagram shows an image of a chloroplast. The image is 5 centimeters long. The actual length of the chloroplast is 5 micrometer. What is the magnification of the image? So in order to calculate magnification, you just have to remember this formula. So magnification is image size over actual size. So in this case, the image is 5 centimeters and the actual length is 5 micrometer. So the difference between centimeter and micrometer is that first you have to know the difference between centimeter and the millimeter, which is 10. The difference between the millimeters and micrometers is, of course, 1,000. So if it's 5 centimeters, then it must be 50 micrometers. So it's now 50,000 micrometers over 5 micrometers, which is 10,000. So here the answer is 10,000, C. So always remember the formula, and the rest of it will be quite easy. Of course, you have to know the differences between the units, but I've written it here, so you remember this one, okay? Okay. Number 5. What are features of osmosis? So osmosis is the movement of water molecules from high to low water potential. And this is important. You always have to say through semi-permeable membrane. Okay? It's the movement of water molecules from high to low water potential through semi-permeable membrane. All right. So let's have a look at our options here. So first one is diffusion is involved, requires cell walls, requires a partially permeable membrane. Like I wrote it here on the right side. Yes, it requires a partially permeable membrane. So semi-permeable membrane and the partially permeable membranes are the same things. So it's either A or C. Does it require cell walls? No, cell walls are only there to prevent the cells from bursting. It has nothing to do with controlling the movement of particles, like water particles or water molecules or whatever. So, nope. Hence, the answer is A. However, let's have a look at the first option here. Diffusion is involved, yes, because osmosis is a specific type of diffusion. So diffusion is the movement of particles from high concentration to low concentration. Okay. So this is the region of high concentration and low concentration here and there. So yes, osmosis is a type of diffusion. So A is the perfect answer. Okay? The diagram shows a apparatus which can be used to demonstrate osmosis. After 1 hour, what would happen to the liquid levels in the glass tubes? So, if you look at this diagram, you can see there is very concentrated sucrose solution on the left side and also concentrated sucrose solution on the right side. So they are both concentrated. It's just that the left one is more concentrated. And outside the solution, or outside the partially permeable membrane, there's water. And well, compared to the solution, water is just water, right? So there's more water in water. Water. You can't say water solution because solution means that you dissolve something in the water. So you can't say water solution. Okay? It's just water. All right? So if you compare to the outside, now there's more sucrose molecules in the solution, so there's less water, which means the water outside will move into the solution. Same thing will happen here as well on the right side. So if the water goes into the left side and the right side of the tubes, then both of them, both liquid level will rise, right? Hence the answer will be D. But this is okay because osmosis will happen, and this was a typical tricky question of using just concentrated sucrose solution. Okay. So since both of them have lower water potential than the outside, they'll both gain water. Hence the answer is D. Okay. Number 7. Which substances are made by linking together glucose molecules only? So, glucose is carbohydrate. And there are several carbohydrates that you should know. Carbohydrates are divided into 3 groups: monosaccharide, disaccharide, and polysaccharides. And in monosaccharides, you guys should know 3 of them, which is glucose, or which are glucose molecules, fructose, and galactose molecules. And what disaccharides are not often asked, so I was gonna skip it, but it's always good to know more, right? So maltose, sucrose, and lactose molecules are within the disaccharide group. And for polysaccharides, you guys should know starch, glycogen, and cellulose. And these guys are made of glucose molecules. Okay, so maltose is actually made of 2 glucose molecules, sucrose is made of glucose and fructose molecule, lactose is made of glucose and galactose molecule. Okay, so you can see that A, cellulose, glycogen, and starch are found here, which all belong to carbohydrates. And like I said before, they are made of glucose molecules. All of them are made of glucose molecules. They're actually made of different types of glucose molecules, but in igcse level, you guys don't have to know that. So anyways, the answer is A. Number 8. When bases pair up in the formation of DNA, what is one of the pairings? So DNA is made of many nucleotides. Nucleotides have, or each nucleotide has base, and there are 4 types of bases. There's a rule called, I suppose you could just call it a rule, but complementary. The rule is called complementary base pairing, which is that the A will always bind with T, C will always bind with G. So here the answer should be B, because C or G will always bind with G or C. So G always binds with C, C always binds with B. So the answer is A. This one was quite simple, right? So always remember this rule. This is called complementary base pairing. Okay? Cool. Number 9. The graph shows the effect of pH on the rate of reaction of an enzyme. What does the graph show? So in the graph, you can see that as the pH gets increased, the rate of reaction also increases. But it also increases to a certain extent, and the highest peak is, of course, where optimum pH for the enzyme activity is found. After that, the rate of reaction will decrease. So in other words, we can actually see this in the other way around, a different view. So which means that the other than the optimum pH, if the pH becomes decreased or increased, the rate of the reaction also decreases. Okay. So what does the graph show here? The rate of reaction. If you look at the rate of reaction, enzyme is not destroyed. It's not destroyed because the rate of reaction is still there. Oops, not that one. So enzyme is not destroyed, and it's not working best at pH 6. It's actually working best at pH 7. The rate of reaction halves as the pH changes from 5 to 7. 5 to 7. Nope. It's actually increasing. Not in half, but a little bit less than half, I guess. So it's not the right answer. D must be the answer. The rate of reaction is same at pH 5. And pH at 8.5. So 8.5 is here. No, actually it's here. Jeez. 다운아, 나 잠깐만 끄고 좀 이따 다음에 택시 타고 전화해도 될까? 그 녹화하는데 소리가 들어와가지고. 아니면 다운이가 마이크 있지 거기. 거기 마이크 음소거를 해주세요. 좋아. 오케이. 나도 내 거 마이크 음소할게. So number D, the rate of reaction is the same at pH 5 and pH 8.5. So at pH 5 and pH 8.5, yep, they do look quite same, right? So the rate of reaction is the same at pH 5 and pH 8.5. So D is the answer for this question. Number 10. The graph shows how enzyme activity is affected by temperature. How can the change in activity between 40 degrees Celsius and 55 degrees Celsius be explained? So 40 and 55 is around, let's say, here. So the activity change between the 40 and 55 is like the rate of reaction is like decreasing dramatically, right? So the right answer cannot be A because first of all, enzyme is not a living thing. So it can be killed. We never say that the enzyme is dead or is killed. And B, the enzyme has been used up? No. As the temperature gets increased, enzyme can actually become used up, which means its active site is occupied, by a substrate. So the enzyme has been used up. No, you can't say that. If the enzymes are used up, which means if they're fully occupied, then it means the rate of reaction is at its maximum point. So at this point, enzymes are all used up. C, the reactants are moving faster. Yes, of course, the substrates, which can also be considered as reactants, they're moving faster, which means they will have more collision, and the rate of reaction will remain high. But you can see that it's not. The activity has halted. It got stopped. So C is not the right answer as well. So D, the substrate is less likely to fit into the active site. Yes, it is right, because the rate of reaction is decreasing, which means that the substrates are no longer fitting into the active site. So D is the right answer. Okay. Probably because the active site of the enzyme became denatured. The graph shows how the rate of photosynthesis of a plant changes with light intensity at 3 different carbon dioxide concentrations. In each case, the temperature is 15 degrees Celsius. So the temperature is the same in 3 different carbon dioxide concentrations or 3 samples. What is the limiting factor for the rate of photosynthesis at point X on the graph? So at this point, it says it's asking the limiting factor, okay? It's asking for the limiting factor. Limiting factors for the photosynthesis is, of course, there are 3 things: temperature, light intensity, and substrate concentration. Or actually, in this case, it must be CO2. CO2 concentration. For photosynthesis, CO2 is a substrate. So, okay, so it's showing that the temperature is remain the same. So you can't say the limiting factor is the temperature because it's not changing at all at this time. So the limiting factors are factors that can affect the life process in such a way that if there's no limiting factor, then it can actually stop the living process, such as photosynthesis. So as you can see here, as the light intensity becomes increased, the rate of photosynthesis also increases. But which means if the light intensity becomes decreased, the rate of photosynthesis also becomes decreased. So to some extent, to this point, the light intensity is indeed the limiting factor. For the second graph, until this period of time or until this light intensity range, not the time actually, my bad. So for this range, the rate of photosynthesis gets affected by the light intensity. So limiting factor is indeed, in this case, the light intensity. I mean, until this position. So the light intensity in this range is the limiting factor for the second graph. For the third graph, the top one, this one, the light intensity is the limiting factor in this light intensity range, because as the light intensity gets increased, the rate of photosynthesis also increases. So light intensity is affecting the rate of photosynthesis here, okay? But after that, the light intensity is no longer limiting factor because it's not affecting at all. For the first graph, of course, the light intensity from this point to here, the light intensity is no longer the rate of no longer the limiting factor because it's not affecting at all. As it gets increased, there's no change. As it gets decreased, there's no change. So light intensity is affecting the rate of photosynthesis only in this part. So yes, light intensity is the limiting factor for this part, but no for the others. No for the rest. Okay. Anyway, back to the question again. So the temperature is remained the same, so we can't be sure whether that's the limiting factor at X. But we're sure that the light intensity is no longer limiting factor at X because it got increased, but the rate of photosynthesis is not increasing at all. So light intensity is not the limiting factor, temperature is not, surface area, you can't find it in 3 factors, right? You can't find the surface area of the plant in 3 limiting factors. So C is not the right answer. That leaves us with A. Okay? A is the right answer. Number 12. The diagram shows a section through a leaf. Which row correctly identifies tissues X and Y? So X is showing spongy mesophyll cell. So the answer is either C or D. Y is called upper epidermis. So cuticle is the cover, is the coating. Okay? And this part is palisade mesophyll cells or palisade mesophyll layer. And this looks like a vascular bundle. So vascular bundle containing, this is the vascular bundle. It contains xylem and phloem. Okay, and these are guard cells, and the hole that the guard cell made is stoma or stomata. Okay. So the structure of the leaf is something that you guys just should know, memorize. The easiest way to memorize it is, of course, by drawing it and labeling it. Okay. Number 13. Which stage of nutrition takes place when food molecules become part of a body cell? So, when food molecules are digested, they become smaller. Okay. That happens through digestion. And when these small molecules are present in small intestine, so this happens in small intestine, actually, digestion. Well, not only small intestine, but quite a lot of the digestion happens in small intestine. So these small molecules, they go into, or they get absorbed into our body, and they go into the blood vessel, and then they travel around our body. And then at some point, the nutrients, the small molecules, will be transported to the cell or body cells. Okay? And this process is called assimilation. So assimilation happens when the substances in the blood vessel gets transferred or transported to the cell and become part of the body cell. Right? So B is the right answer. Ingestion is somebody's just consuming something by mouth. That's ingestion. Absorption is absorbing, like taking in the small substances into the blood vessel. And digestion is, of course, breaking down the large insoluble molecules to smaller soluble molecules. Okay? So B is the right answer. Number 14. What is the definition of chemical digestion? So, digestion, well, we can think of digestion in two ways. One is mechanical digestion, the other one is chemical digestion. Okay? So mechanical digestion happens by a few things. Things like teeth and small intestine. So small intestine, or let's just say intestine, because the peristalsis, so which is the movement of the tube, long tube, inside our body or inside our alimentary canal. So that's a physical movement, and that physical movement, bowel movement, can actually digest the large food molecules. And the chemical digestion happens mainly by enzymes. And the chemical digestion refers to the breakdown of large insoluble molecules into smaller soluble molecules. So A is correct. So the large molecules are often insoluble, and they have to become soluble in order to digest better and also transfer better or transport better. Okay? Because the small molecules, if they are soluble, then they can move around our body when the blood travels. So the blood has a lot of CO2, a lot of H2O, the water, and if these small molecules can become dissolved in the water, as the blood moves, the water will always also move, and the molecules will follow the water. So the A is the answer. Number 15. In which order does water pass through these structures in a plant? So let's draw a tree or plant or whatever. Well, trees are plant. So, okay. The water outside the root, in the soil, they get sucked in, of course, by the root hair cells within roots, and then they get transported to the leaves through a pathway called xylem. And then as soon as it reaches the leaves, it's going to go into the leaves. The leaves have a layer called mesophyll layer. There are two types of mesophyll layers. One is palisade mesophyll layer. The other one is called spongy mesophyll layer. So they enter this mesophyll layer. Hence, the answer should be D for this one. Okay? Number 16. The photomicrograph shows a cross-section through a plant stem. What is the name and function of the tissue labeled X? So, if you look at the structure X, you can see relatively it's got bigger circle than the other ones. So this part, or these parts, are collectively called vascular bundle. So vascular bundle consists of two things: xylem and phloem. The xylems are always concentrated towards the center, and the phloem are placed on the outer sides. Okay, outer sides. So in this case, X is of course xylem, so the answer is either C or D. And the function of xylem is to transport water and minerals. Transporting sugars is the function of phloem. Number 17. The table shows some features of blood vessels. Which row shows the feature of a vein? So there are three types of vessels, blood vessels. The first one is called artery. Artery has small lumen and thick wall. The thick wall has like muscles and elastic fibers. The artery has a very thick wall because it has to withstand a high pressure. Since the lumen is small, the lumen, in other words, is space, is small, the pressure remains high constantly. But for veins are bigger than the arteries. They have thinner walls, and they have bigger lumen. Since they have bigger lumen, their pressure is lower, so they don't have to withstand that much of pressure. That's why the walls are quite thin. And because of the lower pressure, there's always a possibility of blood flowing backwards. So in order to prevent the blood backflow, there are valves. So it closes down, the valves closes down, or the valves close down, my bad, when the blood tries to flow back or flow backwards. So if the passage is blocked, then they have to move forward. So the question was asking the features of vein. So direction of blood flow is always towards the heart. The vein always sends the blood to the heart. The lumen is always large and the wall is thin, so answer is D. Okay? Number 18. Where are valves found that prevent blood from flowing in the wrong direction? So the valves are found in veins because they have big lumen, they have lower blood pressure. So veins contain valves, and there are valves in heart as well. There are valves called AV valve, atrioventricular valve, or valves, and there are semi-lunar valves in the heart, or strictly speaking, it's not in the heart. It's in the arteries like aorta and pulmonary artery, which are placed in or which are connected to the heart. So you can safely say the heart and veins have veins. So the answer is D. Okay? Number 19. The sequence of amino acids in antibodies enables them to complete which function? So sequence of amino acids in antibodies. So antibodies are proteins. And they are made by amino acids. And depending on the types of amino acids, the proteins, the shape of the protein can change, and it also changes the antibodies. Okay. So, what does that mean? That means that the shape of the antibody will change, and the substrates or the antigens, not the substrates actually, antigens that binds to the antibody can vary. Okay. So because of the specific shape of antibody, it will only bind to a specific antigens. So A is the correct answer for this one. It will bind to a specific antigen. Number 20. The diagram shows some of the structures in a human lung. Where is the carbon dioxide concentration highest? So it looks like the blood is flowing from here and it goes out through here. So it says from pulmonary artery. So pulmonary means lungs. Artery means the blood vessel that transports blood from the heart. So B has blood, the blood vessel B has blood from the heart that goes to the lungs. So this is called alveoli. And it provides gas exchange. So it provides oxygen and takes out the carbon dioxide. So B probably will be the blood vessel that has the highest carbon dioxide concentration. So blood flows in order to get rid of the carbon dioxide, and it also enters the lungs to obtain more oxygen. So B has a lot of carbon dioxide first, and as soon as it travels and goes out, the carbon dioxide in the blood has decreased a lot. Okay? So B is the right answer for this one. Number 21. The list shows some processes that happen in the human body. Which of these require energy released by respiration? Okay, so which of these require energy released by respiration? Looks like it's asking about the purpose of energy making by respiration, and number 1 certainly is not the right answer because water entering the cell by osmosis does not require any energy. Only active transport requires energy. So number 1 is not correct. Number 2 is correct. Yes. In order to contract muscles, we need energy. Number 3, impulses travel along neurons. This is very good question. When neuron needs to send its signal to another neuron, it does require energy to send the impulse. So, number 3 is correct, yes. Number 4, oxygen diffuses into cells. No. Both diffusion and osmosis do not require energy, okay? So only active transport. requires energy. So the answer should be 2 and 3. C. Number 22. The table shows the composition of blood entering and leaving the liver and the kidneys. Which row is correct? Okay, so it's looking at the urea in the blood, highest concentration of urea in the blood. Is of course can be found in the blood entering the kidneys. Okay, so the urea, first of all, is made in the liver. Okay, so when urea is made in the liver, it gets transported to the kidneys. So the highest concentration of urea in the blood is found in the blood that enters the kidney. And lowest concentration of urea in the blood, of course, is not the blood that's leaving the liver, because we just said that the blood that's entering the kidneys from the liver has most urea, which is the same thing as the blood that's leaving the liver, right? So A is not correct. Must be B then. Entering the liver? Yes. The blood that enters the liver does not contain urea at the moment. As the blood enters the liver, the things that are inside the blood, especially amino acids, they get broken down and they go through the process called deamination. And produce urea. Okay? So B is the answer for this one. Number 23. Which is the correct sequence of structures through which a nerve impulse passes in a reflex arc? So impulse always starts, or actually not impulse, but the reflex arc always starts with receptor first, okay? So the receptor receives stimuli or stimulus, depending on how many stops are stimulating. So if there are many things that act as stimulus, you could call them collectively stimuli. So stimuli is plural, stimulus is singular. So the receptors, they perceive or they receive stimuli, and then it sends the signal through sensory neuron, and then relay, motor, and effector finally. Effectors are like muscles and other organs. Receptors are like eyes and skins. And, to be more precise, you should say that the temperature receptors or pain receptors or pressure receptors are inside the skin. So these receptors can perceive or receive many stimuli, and then they give this signal. They convert this stimuli into signal and then send it to sensory neuron, which will send it to the relay neuron and then motor, and then effector, and then the effectors can exert its effects. Hence, the answer is D for this question. Okay. Number 24. The diagram shows an eye. Which labeled part contains cells that are sensitive to light? So the eye has two types of cells. Actually, there are many types of cells, but for igcse, you guys only have to know two types of cells. So, which are rods. So, rod cells or rods. The other one is called cone cells, so cones. Okay. And out of these two cells, the cells that are sensitive to light is called the rod cells, or are the rod cells. The cone cells are also sensitive, but it's more concentrated on the colors. Okay. So I would say I'll try to find a layer or the portion called retina. So retina contains both rod cells and cone cells. And there's also another part called fovea, which only contains cones. Okay. So the answer here is D, because the inner part, this part, is called retina. And they contain both rods and cones. So rod cells or cone cells. So the answer is D. Number 25. When hormones are secreted, they affect different parts of the body. The table shows some features that are affected by 4 different hormones. Which hormone is adrenaline? Right. Okay, so this question is asking about the effects that occur by 4 different hormones. So adrenaline is the hormone that gets secreted when your body is in alert mode, like when you're frightened or when you have to fight. It's often secreted. The adrenaline is released by adrenal gland. So this is adrenal gland. And it is actually placed above the kidney. So right above the kidney. So the effect of adrenaline is to increase the pulse rate. So the answer should be either A or C. And it also increases the blood glucose concentration, because, like I said before, it is secreted when you're frightened or when you have to fight. So your muscle has to be ready for running away or fighting. So muscle contraction is needed, and therefore you need a lot of glucose molecules. So your blood glucose concentration gets increased when adrenaline is secreted. And the pupil size has to become bigger, because when you want to fight or when you want to run away, you instantly have to receive a lot of information from the outside world. So the only way that we get, and not the only way actually, one of the few ways that we get information from the outside world is by light, by looking around. So when you receive a lot of light in your eye, then that means you're actually getting a lot of information from the outside world. So you can adapt better, you can react better when that happens. So your pupil size has to increase. So answer already is C. But menstrual cycle, this is got to do with the reproduction. So nope. Sperm production, nope. So when you're about to fight, you're not gonna make sperms, right? Or when you're frightened, you're not gonna make sperms. So if you're male, so both of them are X's, and the answer should be C for this question. Okay. Number 26. When the environment is hot, how do the arterioles in the skin and hair erector muscles react? So, okay, when you're hot, the blood vessels in your skin or under your skin wants to get rid of this heat energy in the body. Otherwise, your body temperature will rise, and that's not good. Okay, you don't want that to happen. You don't want that to happen because your enzymes will denature, and your body is going to become sick. So, the arterioles that are under your skin will dilate. So it's going to increase its size so that it becomes closer to the outside, so it can emit or radiate more heat energy. So dilate is the answer. So the answer should be either A or B. And the hair erector muscle will relax because hair, when you're hot, actually when you're cold, your hair will rise. It will erect because your body temperature, your body will emit heat or radiate heat, and it's going to warm up the air particles closer to your skin. So the function of the hair is to trap all this warm air that's surrounding your body. But when you're hot, you don't want that to happen. So immediately, your hairs will just it's gonna lie flat, not trapping any hot air around your body. So the erector muscle has to relax in order to keep the hairs flat. Okay, so the answer is A. Number 27. The diagram shows one parent, Hydra, growing and releasing an offspring from the side of its body. So which row is correct? So it looks like this Hydra is going on the asexual reproduction. I'm certain about this because it's not involving any other parent, right? It's just doing it on your own. And there's no male or female. So it must be asexual. There's no male or female. There's no gender or sex. So which row is correct? Parent and offspring are genetically identical. Involves asexual reproduction. Yes, it involves asexual reproduction, just like I said. So the answer should be either A or C. And parent and offspring are genetically identical. Yes. The feature of asexual reproduction is that it will only create the offspring that are cloned. So this offspring is exactly the same as the parent. Okay, that's the feature of asexual reproduction. So A is correct for this one. Number 28. Which row describes self-pollination? So self-pollination, well, let's talk about pollination first. So pollination happens when the pollen grains from anther reaches the stigma. So this is the stigma. The stigma of the female part of the plant. This is the anther, which is the male part of the plant. And these are pollen grains. Okay. So pollens transferring from anther to stigma is pollination. But if the pollens from one plant travels to the stigma of the same plant, then it's called self-pollination. So different flower on the same plant. Yes, this is a trick one. It says different flower on the same plant. So although we see a lot of flowers, like a lot of pictures like this, and we immediately think that, okay, there's one flower per plant, but that's not true. In one plant, there can be multiple flowers, but they are exactly same things. So exactly same organisms, or it's like they're like clones. Okay. So, but this one, a flower on a different plant of the same species, no. Nope, self-pollination is not. It's not the movement of pollen from anther to stigma of a flower on different plant of same species. So it sounds a bit ambiguous and it's quite confusing. This means same species, okay. Different plant of same species mean they are the same species, but they are different flowers or different plants. So they have exactly different genetic information. They have different genes. So the second one means that the pollen is moving from here to here. This one refers to this. So it's not self-pollination. This is cross-pollination. Okay, so same flower, yes. So B is the answer for this one. The middle one is not the right description for the self-pollination, but these two are. Okay, so B is the right answer. Number 29. Which feature is only found in the male gamete? So male gamete means male, it's like a sperm. In plants, they're pollens, but for animals, male gamete is a sperm. So if you look at the structure of the male sperm, then it has acrosome, nucleus, a lot of mitochondria here, and a tail. This is called midpiece. And this is acrosome, this is nucleus. Okay? So only found in male gamete? Looks like acrosome here. The jelly coat is present in an egg. Enzymes are present in both because they're both cells. Mitochondria is present in both because they're both cells. Okay? So A is the right answer for this one. By the way, the acrosome has a function in let's say breaking down the jelly coat like this. Okay, so this stuff or the stuffs inside the jelly coat, not the jelly coat, acrosome, are enzymes that breaks down the jelly coat so that the nucleus can enter the egg, and that process is called fertilization. Okay. Anyways, the answer is A. Number 30. The graph shows how the mass of DNA changes during a mitotic cell division. Where on the graph are two cells formed? So, mitosis happens after the cell replicates its DNA. Okay, so this is called mitosis, but in order to happen, DNA has to become replicated. So this is mitosis. Okay. So as you can see, it says mass of DNA per cell starts at this level, and as time goes by, it stays still. But then at some point, the mass of DNA per cell increases, right? So this is where B is when DNA replication is happening, and then it remains the same for a while, and then the mass of DNA per cell suddenly drops. So this is when the mitosis is happening. So I'll just draw it here. Let's say there was one chromosome, one strand of DNA. When DNA replication happens, it becomes 2. And then during mitosis, it becomes 1 again per cell. So let's say the mass was 10 grams, which doesn't make sense anyway, it doesn't make sense at all. But let's say it's 10 milligram, okay? It becomes 20 milligram and then 10 per cell. So this is 10 and that's 20 and then it becomes 10 again. That's when the mitosis happens. So D is the right answer for this. Okay? So this is here. Replication happening here. And then this is here. And this is when mitosis happens. So, all good? All good. Number 31. What happens during meiosis? So meiosis is basically it's a cell division to produce sex cells, like a sperm or egg. So meiosis is quite similar to mitosis. However, there's a huge difference. It divides twice and creates 4 cells instead of 2 cells in mitosis. So in mitosis, it just happens. Cell division just happens once. And what's more important, mitosis creates clones, whereas meiosis creates genetically non-identical cells. So genetically, they're all different. As per mitosis, they create all clones. So, what's the difference? And also, since they produce exactly the same clones, the chromosome numbers are the same as well. So the product of mitosis is always diploid. So cell always starts from a diploid, and then during mitosis they produce, their clones. So they should be diploid as well. But for meiosis, these guys are sex cells. So they should have, in humans, they should have 23 chromosomes. So they are, haploids. Okay, so 2n turns into n for meiosis. So 2n means there are pairs of chromosomes, so it's called diploid. N is, they're not in pairs anymore, they're singlets, so they're called haploids. Okay, so during meiosis, what's happening? The diploid cell divides to form haploid cell. So B is the correct answer. Number 32. Red-green color blindness is a condition that occurs more frequently in men than in women. Which statement about this condition is correct? So red-green color blindness is called sex-linked disease. And more specifically, it's X-linked, meaning that the gene that causes this problem or this disease or this disorder is only present in X chromosome, not Y chromosome. Okay, it's only present in X chromosome of the sex chromosome. So for females to have this red-green color blindness, they would need to have two recessive genes. If they have one dominant allele and one recessive allele, they are called carriers. They're normal, but they carry the gene that can cause the color blindness. If they have two dominant alleles, then they are normal. As for males, we have XY chromosomes. So, for low battery now. Okay, so for males, if they have dominant allele on their X chromosome, then they are normal. But if they have recessive allele, then they are affected. So poor males, right? Poor buggers. So females have, 1 in 3 chance of having the color blindness, but for males, it's like half, 1 in 2. So this is why, statistically speaking, there are more males with color blindness than females. Okay. So number 32, the answer is B because it's a sex-linked disease or sex-linked disorder. Number 33. Which statement explains why the allele for sickle cell anemia is commonly found in human populations in certain parts of the world? So sickle cell anemia is a strange genetic disease or genetic disorder. It's caused by HBS allele. If you have HBA, or two of them, you're normal. So having two of these, you have normal red blood cell. If you have 1A, 1 and 1S, then you are not a carrier, but like half of the red blood cells are normal and half of them are sickle-shaped red blood cell. If you have 2 S's, then your all of your red blood cells are sickle cells. Okay. The funny thing is that, or interesting thing about this is that malaria can only attack the red blood cells that are normal. For the red blood cells that are turned into sickle cells, they're safe from the malaria. And of course, malaria are transmitted by the mosquitoes. And this is why some parts of the world has a lot of sickle cell anemia genes left in the population. Because normal people, they get affected by the malaria and they often die. For those who have sickle cells only, they can't actually, they can't live like when they're born. So when they're embryos, before they're born, they often die. So these two are quite hard to survive in some parts of the area or in some parts of the world. And these guys can live because half of them are normal, half of them are sickle cells. So even if the normal cells get affected, they still have sickle cell anemia. I mean, they still have sickle cells. So they get protected by, or against it, actually. They're against the malaria. So people with sickle cell anemia gene, like half of them, get protected against the malaria, and that's the answer for number 33. Okay. So in a way, sickle cell anemia is like co-dominant disorder. Because if you have two of each, actually one of each, if you have both A and S, then they do both show their characteristics. One is the normal hemoglobin or normal red blood cell. The other one is the sickle-shaped hemoglobin or the red blood cells. So, okay. So when this is affected, this one still survives, and they can still transport oxygen. Like not much, but yeah, they still do the job. So, okay. So the B is the answer. Question 34. The development of antibiotic resistance in bacteria is an example of which process? So development of antibiotic resistance in bacteria can be caused by, first, if there is a variation in bacteria. So amongst all the like millions of bacteria, there can be one bacteria that has antibiotic resistant gene. So that's called variation. And second, if antibiotics is put into the region where there are a lot of bacteria, including that specific bacteria which has an antibiotic resistance, it's going to give like a lot of struggles to them. And then during the struggle, that specific bacteria will adapt and it will survive. Okay? That means others will die, but it will survive and it's going to reproduce itself and it's going to pass their genes or pass its gene to the next generation. Okay? And that's a development of antibiotic resistance bacteria overall. So, and these steps or criteria are the example of natural selection. Okay? Evolution by natural selection. So B is the right answer for this one. So when we talk about the natural selection, it is very crucial for you guys to mention about these criteria, like in this order. So if you can mention these in order properly, then you're gonna get like full marks easily. All right. So this is very important. So keep in mind. All right. Number 35. The diagram contains information about the number and mass of organisms in a food web. What is the total biomass of all the primary consumers in this food web? Okay. What is the total biomass of all the primary consumers in this food web? Okay, so we just have to figure out what the primary consumers are. So hazelnuts are, of course, producers, right? Producers are often plants. Hazelnuts are plants. So, and the next one that eats hazelnuts is, of course, the primary consumer. So, squirrels and shrews. So, if there are 150 squirrels, and if there are 3,500 shrews, we can do the math, right? So it's pretty simple maths. So that's going to add up to 130 kg. Okay. So the answer is A. Number 36. The diagram shows some of the stages in the nitrogen cycle. Which arrow represents the process of nitrogen fixation? So nitrogen fixation is fixing or using nitrogen gas in the air. So it's converting to the nitrates. So A is the right answer. Let's have a look at the others as well, just for fun. Okay. So it looks like the plant is getting eaten by herbivore. That's like digestion or ingestion. Not digestion, but probably ingestion or consumption. And if the dead plants become dead organic matter, that's called decomposition. And they become nitrates, and this process is called nitrification. So dead organic matter, like ammonia or urea, if they become nitrates, then the process is called nitrification. And if the nitrates become nitrogen gases, that process is called denitrification. Okay. So anyway, this question is asking which process is nitrogen fixation. So the answer should be A. Number 37. Which term is used to describe the phase of fastest growth in a population of bacteria? So when we talk about the population graph, it shows a sigmoid curve. And there are different parts in this sigmoid curve, which is it consists of lag phase, log phase, and there's stationary phase, and there's also death phase. So log phase means exponential, and of course the fastest growth in population is shown by exponential part, right? The log phase. Hence, the answer should be C. Okay. Stationary shows no growth. Lag phase shows very slow growth. Exponential shows very fast growth. Sigmoid is the name of this graph, or name of this shape of the curve, sigmoid. All right. Number 38. Which are reasons why bacteria are often useful in biotechnology? So bacteria are quite useful for us because they help us to make what we need. And they can help us because their genetic codes are the same as ours. What I mean by that is that when we look at DNA sequence, if the DNA sequence, like a part of the DNA sequence, is like A, C, T, T, A, G, C, T, A, C, blah, 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 blah. And let's say that this sequence, or this codon, A, C, T, represents a certain type of amino acid, okay? If we put this codon into bacteria, it's going to make exactly the same amino acid. The same amino acid that's made in our body, right? So which means that the genetic code is the same. They get interpreted the same way. So if I want to make a human insulin, I'll just put in the human insulin gene into bacteria, and they will just make the human insulin for us. So number 2 is very crucial. And of course, one, lack of ethical concerns is very important as well. So we can just use bacteria because there's lack of ethical concerns. We don't care, right? I mean, there are living things, but the scientists don't really care about that. So, yeah, which is, in a way, quite sad, but yeah, well, that's the way it is. So, 1 and 2, let's have a look at 3. Their DNA is located in the nucleus. No, bacteria do not have nucleus. Okay, so number 3 is not correct. So A is the right answer for this one. Number 39. Penicillin is produced in a fermenter by growing the fungus Penicillium. The graph shows how the mass of living penicillium fungus and the concentration of penicillin changed over time. When is the best time to collect the penicillin? Okay. This question is quite interesting. So when is the best time to collect the penicillin? So looks like the concentration of penicillin is at its highest in day 5, right? So what we want is not penicillium, but we want penicillin. So we want the highest concentration. So day 5 is correct answer. Okay. So the way that you read this, or interpret this graph is that the mass of living penicillium fungus is shown with this straight line, or not the straight line, but this normal line. So you read it on the left with the left y-axis, but then dotted one, the dashed line, is showing the concentration, and you read it with the right side, the y-axis on the right side. Anyways, you want to get the highest concentration, so day 5 is the correct answer for this one. Okay? Number 40, the last question of this paper. The number of Atlantic bluefin tuna fish found in the Atlantic Ocean has significantly decreased in the last 50 years. Which method would make the biggest improvement in the conservation of tuna fish stocks? Of course, in order to conserve tuna fish, what we can do is, A is not correct. You have to increase the size of the holes in fishing nets so that the young ones can just escape and we only catch the bigger ones. Genetic engineering of tuna? Not sure about that because this is rather very vague. So we want to conserve. We don't want to change the tuna, right? Number C, introducing fishing quotas. Yes, if we set the number of fishes that can be number of fish or number of tunas that can be caught in a day or month, we can certainly conserve the number of tuna. Okay, selective breeding of tuna, no, it's going to create another type of tuna. We're not conserving it. Okay, so C is the right answer for this one. We're good.